Hello, my name is Samantha Northrup, and I did my research project on Bacillus anthracis, more commonly known as anthrax. Anthrax is important because it's considered a bioterrorism weapon, potentially. Coming into contact with anthrax can be very deadly. It is one of the reasons that the CDC classifies it as a Category A agent and is a biosafety level 3. Anthrax is a hardy spore. It is extremely resistant to natural conditions and is capable of surviving for many years in multiple environments. Bacillus anthracis is classified thus: domain, bacteria; phylum, Firmicutes; class, Bacilli; order, Bacillales; family, Bacilliaceae; genus, Bacillus; species, B. anthracis. Bacillus anthracis gets its name from the Greek word B. anthrachis, meaning coal, because of the black lesions that develop on the skin after a cutaneous, or skin-level, exposure. This bacterium was actually the first one discovered to cause disease, also known as a pathogen, by Robert Koch in 1876, and was the purpose of the first vaccine containing live organisms developed by Louis Pasteur for his veterinary anthrax vaccine. Anthrax causes anthrax poisoning, which can lead to many other diseases. It is not communicable, which means it does not spread from human to human, so contracting it means that a person must have come into contact with a spore at some point. It can be inhaled through spores in the air, ingested through spores in food, or infected through a cut in the skin. It is possible, though only slightly, to contract anthrax poisoning from another person if their skin lesion comes into contact with the broken skin of an uninfected person. For my peer-reviewed academic journal article, I chose one that goes into the specifics of what makes anthrax so dangerous. The hypothesis of the scientists in the articles was complicated, but it simplifies to which medication can be used to the best effect in the event of anthrax poisoning, and also to see how quickly B. anthracis develops a resistance to said medication. During their research, the group of scientists grew B. anthracis in a sporulation growth medium and then exposed it to 3 quinolones, or antibiotics, and 3 macrolides, also called bacteriostatic antibiotics. The evidence following the experiment suggested that the microbe showed some slowing of growth, but still maintained a hold in the bacterial plate. This table and the conclusion in the article suggest that during long-term treatment with any of the antibiotics, B. anthracis would develop a resistance to them. This proves their hypothesis that anthrax is highly dangerous and is resistant to most antibiotics given enough time. Anthrax poisoning would be classified as re-emerging, as it appears every so often across the globe. While it is uncommon in the developed world, for example, less than 5 cases in the United States each year, anthrax is still dangerous because of its potential use as a bioterrorist weapon. Between 2,000 and 20,000 people are infected with anthrax each year. Anthrax poisoning is diagnosed by culturing suspected infected blood, respiratory secretions, or skin lesions, or by testing for specific antibodies. There are many symptoms of anthrax poisoning, including bloody vomit or stool, fever and chills, swollen glands, painful swallowing, black skin lesions, and hoarseness. Inhalation anthrax, which is the deadliest, can begin showing symptoms after 2 days and last for months. The proper treatment for this varies, as multiple antibiotics may be given in an effort to stave off resistance. Given the proper treatment as quickly as possible, about 55% of patients survive the infection. The pathogenicity of this microbe involves its protective endospore layer and the protein capsule, which is the key to the invasion of the immune system involving plasmids.